Okay. So I think I'm gonna, I think I'll probably start and let people come in as the talk is going. So Xihong, people usually trail in, you know, in the first 5 minutes or so. Okay, so I'd like to welcome everyone to our, we'll call it the series finale for our Decoding Pandemic Data seminar series here at Brown. I'm really pleased to welcome Professor Xihong Lin. Xihong is a longtime friend and colleague, and we were just discussing before we opened the waiting room that we were collaborators on a paper together. I feel really lucky to have been one of your collaborators, Xihong, and our community in biostatistics, and I think in the broader community in data science, views Xihong as really one of our true scientific and academic leaders and role models. So Xihong was formerly the chair in the Department of Biostatistics at Harvard. She has been the leader of the Committee of Presidents of Statistical Societies. She is a member of the National Academy of Medicine, and she's also been awarded the Mortimer Spiegelman Award for that's given to a statistician making seminal contributions to public health. And this is just an abbreviated list of Xihong's accolades. She leads a number of large center grants looking at population genetics and genomics. And she recently, to nobody's surprise, jumped right into the fray when COVID broke out. And she has really used her data science prowess and her statistical training and expertise to unlock some really complicated data sets, particularly related to the initial outbreak in Wuhan and more recently to the larger COVID pandemic here in the US and around the world. So Xihong, you know, it's really an honor to have you speak at this series. It's really great to see you again. So grateful that you said yes to this invitation, and we're really looking forward to hearing what you have to say. So welcome.
And thank you so much, Joe, for the very kind invitation, also the introduction. And so I visited Brown a couple years ago. It's really wonderful to see how the Department of the Biostatistics grow. So it's wonderful to see this data science initiative at Brown. And so so I will share with you some of the work my group has been doing in the last few months on COVID-19. So let me share the screen. Okay, so I will first start from the Wuhan data, and then we'll talk about the US data and also the data in the world. So this is the curve I just downloaded today. So you can see that right now there are over 64 million cases and also 1.5 million deaths around the world. So if you look at the number of cases on the in the curve on the left, and then you can see that the US has the bimodal curve, and then right now look like the European Union and also UK, the curve has been banded, and also especially the France has been doing really well, and because the control measures they recently launched in the Europe, and but if you look at Sweden, as you know, they used a different strategy and looking for the herd immunity. You can see that Sweden, the case rate has been going up, unlike the European countries, and but the China is in the bottom here. And similarly, this is the number of death curves, and then you can see the curve is very similar patterns. And so the France, because their control measure recently launched, you can see the curve has been banded. But Sweden, Sweden and also the European Union and also the US, and the number of deaths has been going up. But recently, the number of deaths in the US has been going down slightly, and China has been doing really well as well. So let me start from the historical perspective. And so I first worked on COVID-19 really by coincidence. This happened in mid-February. And so the Chaolong Wang was my former postdoc. And so he after he spent some time in Singapore, and then he went back to China and joined the faculty in the School of Public Health at the Tongji Science and Technology Universities. And so in mid-February, I wrote him a text ask how he and his family was doing, because Wuhan was epicenter. And then he was telling me that he and his colleague were analyzing the Wuhan data. And then at that time, there was already one case in Seattle and also one case in Boston. So I got I got a sense that it could be an epidemic in US. So I decided to join the force and working with them, analyzing the Wuhan data. So the team worked really hard and then analyzed the data of 26,000 Wuhan cases and until February 18th, and with the goal of sharing the findings with the world as soon as possible to help the US and other countries. And then we posted the preprint on March 6th. And so this preprint got a lot of attentions, and you can see the number of the download and also abstract views. And also I summarized the findings on your Twitter. And then the amount of the material was too much in this preprint. And then we decided to split the paper into two parts and also by adding in additional material. And so this resulted in two papers, and one was published in JAMA in the mid-April, and another was published in Nature in the summer, in July. And so An Pan and Chao Long, they were both formal Harvard School of Public Health alums. And so Tang Chunwu is the Dean of the School of Public Health and as the Huazhong Science and Technology Universities. So I will show you what we find in those papers. And then, so this work got quite a bit of attention in the media and also in the government. So you can see those are the media coverage and also interviews. And also in March, I decided not to do any interviews, and because as about statisticians, and I try to avoid the media. But then in April, I decided that it's a good idea to talk with the media, and so to help the media and the public to understand the findings better, so the effective strategy and can be implemented to help US and other part of the country. And one thing I learned is when one talk with the media, it's very important to speak in simple language without the jargon, and then people can understand. And then I also was invited to serve as a witness in the Science and Technology Committee of the UK Parliament on April 17. And so this committee has about 8 to 10 parliament members. It's like the senators and in US. And then the committee later on in May, they wrote a letter to the Prime Minister Johnson and made 10 recommendations. And several of the recommendations I made during the sessions and were included and in their letter to Minister Johnson. So it's nice to see that statistician can help the community and using the data fight against the COVID. So let me start from the major findings of the Wuhan data analysis. So let me first introduce this RT. So this is called the effective reproductive number. So many of you probably are now familiar with this concept. And so basically this measures the degree of spread. So if RT greater than 1, so you can see in the figure on the right is RT equal to 4. So that means one person can infect 4 people. And then you can see this become spread quickly. And so in order to control the epidemic, one need to make this RT less than 1. And so then the epidemic will be controlled. So this is how the Wuhan data look like. And so in the paper we published in JAMA and Nature, and so we update the analysis using the data up to March 8. And so you can see that before January 23rd, and there was no interventions. And then you can see the number of cases went up quickly, exponentially. And then on March 23rd, sorry, on January 23rd, that was the launch of the lockdown. And then the people were asked to stay at home. And then you can see the RT curve, and the number of cases rising rate also went down. So then, but it's still increasing. And then on February 3rd, that was the launch of centralized isolation quarantine. So the city built over 16 field hospitals. And then you can see the number of cases dropped very quickly. And on the February 17, that was the launch of the universal screening. So basically the community workers and went to every single household to check whether people has any symptoms. And then after that, you can see the case further drop down. And by March 8th, and then the RT value got really low. Then you can see the on the right, this shows the RT estimate in each of the 5 periods. So before the intervention, before the January 23rd, the RT was about 3.5. So that means that one person infected 3 to 4 people. So this tells us the first feature of COVID is this high transmissibility. And then after the lockdown, so they basically launched the social distancing, had people stay at home. And then the RT dropped dramatically to a little above 1. So that is not good enough. And because I'll explain that because there's the social distancing help reduce RT, but because the family transmission is common, so this could not control the family transmission well. And then after centralized isolation and quarantine, you can see the RT drop dramatically, and by March 8 is about 0.27. And then the city was, so by March 18, and there was 0 infected cases observed, confirmed infected cases. And by April 8, the city was reopened. And so we analyzed that. So how did we estimate those RT? So we basically extended the traditional SIR model, and by introducing a few components. And so this part is this is a susceptible cases. And then from the susceptible cases and to symptom onset, this period called incubation period. And so from the exposure to the pre-symptomatic period. So this is what we introduced and to model the COVID-19 data. This is called the latent period. It's about 3 days and in this latent period. And from the pre-symptomatic period to symptom onset, and so this is about 2 days. And so during the latent period, and even a person was infected, but a case was not infectious. But during the pre-symptomatic period, and the case become infectious, could infect others. And so therefore it's important to capture those cases before a person has a symptom. And then because the limited testing at that time, so not every single case was ascertained. So therefore we introduced additional a compartment. So this I is ascertained cases, and this A indicate unascertained cases. And so we built the differential equation model and included in the Poisson regression. And so in this model, the data are here. This uncertained cases, this basically is the case, those are the data point here, those are the I's. So those are only the observed data. All the rest come from the differential equations. So by fitting this model, so then those give us the RT estimate. And so we have divided the data into 5 periods and then allow the RT change over the time or over this period. And then from these calculations, and the first takeaway is from analyzing the Wuhan data, we found that social distancing is important, but it's not good enough. It helped reduce R to be close to 1, but it's not below 1 yet. And the reason is social distancing help block the community transmissions, but within household transmission and also close space transmission still important. And so those are common. And so by adding the centralized isolation and the quarantines and to social distancing, that helped quench the curve and stop the epidemic in Wuhan, as I mentioned, and they had a 0 confirmed case by March 18th, and then the city was reopened on March 8th. This whole period was it's about just 2 months, 2 months-ish. And so it's important to see whether the result could be replicated in other countries. So those are the data from Italy and in the spring, and also in Germany. As you can see that in both Italy and Germany, they also use the social distancing that helped reduce the R to be later around 1, and but could not be dropped below 1. And so those results basically were replicated and in other countries. And the second feature we found was that because we have this A part, that module unascertained cases, and we found that the second feature of COVID is highly convertedness. So we showed that about 87% of the cases were undetected. So this red part is the detected cases, and this yellow part is undetected cases. And so those undetected were unascertained cases were mainly asymptomatic or mildly symptomatic cases. And then those blue part is the pre-symptomatic cases. And so we estimated the total prevalence is about 2.5% in Wuhan. And so what that tells us is the detected cases are only the part of the top, the tip of the iceberg. And so even those asymptomatic cases were mildly symptomatic cases, and so they were not detected, but this could still infect the others if was no control measure. But in Wuhan, because there were control measures, and so that helped prevent the asymptomatic and mildly symptomatic cases from infecting others. So this would basically tell us that because there's a large number of undetected cases, and so therefore one need to be careful and when we consider reopen. So we showed that if one reopen too early, so basically if one lifts the restriction after all the restriction, after the first case of first day of 0 case, and in 14 days. And so basically if one observes the 0 confirmed case and then reopen in 14 days, then there's 97% of chance of resurgence. And but if one observes the 0 confirmed case for 14 consecutive days, so that basically 0, 0, 0 for 14 days, then one reopen, then the resurgence probability is 32%. So as this result basically replicated in US as well. And so in US, as you recall, the CDC reported the total number of cases were about 6 to 24 times of the number of the detected cases. And also, as you recall, in May, and the South opened early, and then we saw the resurgence. So therefore, one need to be careful and when one reopen and lifting the control measures. So the take home number 2 is, so when the single intervention was not sufficient, one need to have multifaceted intervention, including the mask wearing, social distancing, and widespread testing, contact tracing, and supported isolation, quarantine, and also the vaccine. And so this can be illustrated by this sweet cheese model. So that means one need to use multiple interventions simultaneously. So if one just has a testing, it's help but not good enough. One has a mask wearing, it help but not good enough. And so one need to provide the other control measures such as supported isolation and quarantines. And also vaccine is not a silver bullet. And so And so the vaccines right now is really wonderful news. Both the Pfizer and also Moderna phase 3 trial has 95% efficacies. And today the UK government and just approved and issuing the approved the Pfizer's vaccine. But this vaccine will not be available to everybody. And also one need to not even with it available, and not everybody is willing to take it. So still a lot of work to do. And also, we to help combat COVID, we also launched this RT map website. And so this provides the country level RT values at any given time, and also the state level, and also the county level. And so here is the country level. So I just downloaded from this website we built in the spring. And so you can see that Europe seems like doing quite well. In terms of RT, the curve has been banded, and because several countries launched the lockdown recently. But the US, you can see the curve has been going up, but recently it's getting a little bit better. And so this we also provide the state level and RT map. So this is the most recent RT estimate. So this is Rhode Island. So you can see the Rhode Island right now, the RT is a little above 1. And so then, but the middle part of the country, it seems like the cases has been going down. And so we also provide the county level RT map. So this is the several counties and in Rhode Island. So you can see that Providence is here. And so look like RT is pretty similar across different counties in Rhode Island. And so you can see in the spring, and RT value was quite high, and but then has been going down, and especially in the summer, it's been doing really, really well. Many of the counties in Rhode Island basically have very small number of cases. And but recently in the fall and also in the winter, you can see that the number of cases have been going up. And also, this figure, this map shows the county-specific death rate comparing the spring, summer, and fall. You can see in the spring and mainly the northeast and also the southwest, especially the Native American reserve areas were affected most. And in the summer, it's mainly the south and also the southwest. And now you can see in the fall and the winter, it's went to the Midwest and rural areas. So what are the risk factors associated with infection? So we look at both the Wuhan data and the US data. So in the Wuhan data, so this was published in JAMA. And so as you can see that for this, we calculate the case rate and for different age groups and in different period. And then you can see the older people, and they had a higher risk of infection. On the right, and this compare the case rate comparing the gender groups and also healthcare workers. And so you can see male and female, they have a similar case rate in Wuhan, but healthcare workers, they had a higher infection rate, especially before the intervention. And so we also launched an app in the spring, and we call the How We Feel app. This was in collaboration with Pinterest and also my colleague Feng Zhang at the Broad Institute. He's most well known for CRISPR gene editing. And so this app has already over 750,000 users and also 40 million responses. And so in this app, and we collect the information about the symptoms, behavior, testing result, and the comorbidities and others. So this paper was published in Nature Human Behavior and in the summer. So here are the major findings. And so first, who were most likely to be tested in US? As you can see that the people with CDC symptoms, and they were more likely to be tested. And also healthcare workers and essential workers, and also people of color, they were more likely to be tested. And so this makes sense. So that means the people who are tested, those are biased samples. And now we want to see what factors associated with the positive PCR test. Because the people who were tested and were biased samples, so in the analysis, we need to account for the biased sampling. And so we use the IPW method to correct for the selection bias. So these are the major findings. So the first, you can see that the males has a higher risk of infection than females. And the second, and people of color, especially Hispanic and African American, they had a higher risk of infection. And also the essential worker, healthcare worker, and they had a higher risk of infection. And secondly, under the household exposure and the community exposures are high risk factors and for infection. And so you can see that with the household exposure, the odds ratio of infections is almost 17 times. And then the odds ratio of community exposure is 3 times. And so this result is very consistent with the Massachusetts data and also the CDC data, which were recently released. In Massachusetts data, it shows that 88% of the COVID cluster are household. So this result also are consistent with the Wuhan result. So that means the transmissions happens more within the household and within the families, the gatherings. And also, what are the most important symptoms? And we found the most important symptoms are the loss of taste and smell. And so the odds ratio is 33. And so that's very high. And so we found among those who had a PCR positive test, 40% of them has loss of taste and smell symptom. And among those who were not tested, only about 0.6%. And this symptom was much more predictive compared to fever and cough. And also this symptom helped because this symptom is pretty rare symptom. And so this helped distinguish from the flu symptoms. So this take-home message number 3 is it's important to remain vigilant. So to keep up the control measures and also help with the vaccines. And in particular, it's important to protect vulnerable groups such as healthcare workers, elderly, essential workers, and also in order to, from the Wuhan data, and so we learned that important to go for zero COVID. And so what that means is we need to reopen when the number of cases are sufficiently small, and also with the control measures still on. So as you recall, in the May, and the South opened too early, and then there were resurgence. And also because there were many asymptomatic and mildly symptomatic cases, and so therefore those control measures need to stay on, even reach the zero confirmed case. And also resurgence is likely to happen if one reopened too early and if one lifts all the measures too quickly. And so also the death is not the single endpoint. And it's important to consider the long hauler effect. And so think especially for the young people and who, as you recall, like in the spring, and the more elderly were infected, but in the summer and the fall, more young people were infected, and then death rate went down, and in the summer and also in the fall. But the young people could still, even the death rate, because more young people were infected, so that's why the overall death rate have been going down. But because one still need to pay attention for the long hauler effect. So and also the associated the comorbidity measures, and such as blood clotting. And also even though the vaccine has been really successful, and so that both from the Pfizer's effort and Moderola's effort, and however, there need to be a lot of work on the vaccine uptake and also educate the public to take the vaccine. So there's still a long road to go. So finally, and to really fight against the COVID, and so we have to stay together. And so then also important to let the data speak and also develop evidence-based strategies. And also we show that the 2 features of the virus, one, it is highly transmissible, and the other is highly convert. So that means that many cases were undetected. So the widespread testing is important. And also single intervention measure is not good enough. It's important to use multifaceted intervention measures, and also it's important to develop strategy to enhance implementation and also compliance. And those are very critical, the implementation and also the compliance. And also it's important to detect the asymptomatic and mildly symptomatic cases early on, and then they help them with supported isolation quarantines, and then that will help reduce the spread. And also important to remain vigilant and reopen slowly when the number of cases is small, and also keep the control measures on. And so this is really a joint work, and with many colleagues in Wuhan, many student postdocs in my lab, and also many colleagues and in the How We Feel team. And thank you.
Xihong, thank you so much. I was just trying to get my computer set back up. This is, it struck me as you were talking that this is a fabulous synthesis for the semester because it touches on a lot of different themes that we've seen in individual talks. You know, modeling and use of apps for reporting back symptoms, also some epidemiologic content, really pretty remarkable. So, well, I do have at least one question that's come in, and there may be others as we get going. So the first question is from Tom Seguros. I hope I'm pronouncing your name right, Tom. And I guess he's made himself visible and has a question for you. Go ahead, Tom.
I would just be interested to hear more about how the how you develop the estimate for the non-ascertained cases.
Yes.
And Tom, sorry to interrupt. Could you just tell us where you're coming from?
Oh, I'm sorry. I'm in the computer science department here. I'm research faculty.
Great, thank you.
Great. That's a great question. And so as I mentioned that we use this Poisson partial differential equation transmission dynamic models. And so this is the partial differential equation and which are built to model the data. The data are here. This basically is the uncertain cases. Those are the data. So basically this is the data. This is what we observe. And then by using those data and then through the partial differential equation, and one can estimate A. And so this A, this indicate uncertain cases. And so as you can see that from the fit, you can see this is basically the dotted curves, and you can see the fitted curve work quite well and for Wuhan data. And we allow the RT and also the uncertainty rate and the change from one period to the other. And the model was fit using the Bayesian approach, MCMC.
Thank you.
So perhaps a follow-up on Tom's question. So in order to identify...
Oh, sorry, Tom, just say where you're coming from, sorry.
Oh, hi, my name is Tom Tricalinos. I work here at Brown Health Services. So in order to identify the unascertained, you know, the identification will come through priors, no? So you allude to the fact that this is being obtained by solving the differential equations, and that is fine. But really, you cannot identify that part unless you have informative priors. Isn't that correct? I mean, this is what we see in our in a similar model that we have for Rhode Island.
Yes, I think that's a very good point. So let me go to here. Then definitely, I think one needed to make assumptions. So any of those dynamic transmission model need to make assumptions, and we make assumptions as well. So those assumptions come from external data. So for example, like we need to assume in this situation, we need to assume that incubation period. What is the incubation period is, and so that is from the external data. This is about 5.5 5.2 days. And also we need to assume that at the initial period, and what is the ascertainment rate at the so like here. So in the initial period, what is ascertainment rate? And so in order to estimate the initial ascertainment rate, so we use the flight data from Wuhan to Singapore. And because of Singapore had very good testing at that time, so they basically for all the user passengers data flying from Wuhan to Singapore, we identify how many people were ascertained. And so that gave us the initial ascertainment rate. So as long as we have some kind of, that's one point, we have some kind of initial estimate, then we will be able to estimate ascertainment rate from using the partial differential equation for all the period.
Why did you say partial differential equations? These are ODEs, no?
Yeah, so you can see this is partial differential equations. So those are so basically the data are modeled, those are the means in the Poisson. So here is the data. So here's the data.
Mm-hmm.
And so the data, we follow each data point, follow a Poisson distribution with a certain mean. And this mean is basically the I part. And then this I, because we have so many compartments, right? And so the I is related to other compartments through this partial differential equation based on transmission dynamic.
Joe, I have a couple of questions that are kind of like technical, and perhaps I could be sent to a paper or so, but dare I just float them?
There's a couple questions in the queue, so why don't I deal with those first,
Sure.
and then cycle back, if that's okay?
Yeah, I will be happy to get your paper. That would be great. Yeah.
Okay, so Allison DeLong has a question. Ali, do you want to ask, or do you want me to ask?
I can ask. So
Just tell us where you're coming from.
Oh, I'm at Brown in the Department of the School of Public Health in the Center for Statistical Sciences. So my question was, is there a way to use your modeling framework to suggest the best populations to vaccinate first when the vaccine is limited?
Yes, so let me find. Oh, here. Okay, so basically, when the vaccine should give to the vulnerable group, right, first. And so if you look at the US data, you can see that the elderlies, and so here, if you look at the US data, then the people, the healthcare worker and essential workers, and they are at higher risk of infection. And so, and also like, because this data was, if you look at the spring, because this is the cumulative data, if you look at the spring and the Wuhan data, elderly, they are at higher risk of infection. So that's why that the vulnerable group, and because they are higher risk of infection, then they should have priority and for having the vaccine first.
Thank you.
I think this is what's recommended by CDC and also National Academy of Medicine task force.
So this has the outcome of being infected versus getting very sick and dying, I guess, too. Like you're.
Well, yes, the data here is this data here is the infection data, and also in Wuhan data we also have the severity result, and so I did not include it here. So for the severity result, we found that older people are much higher risk of becoming severe.
So are you, Ali, I don't know if this is what you're getting at, but I would think with vaccine distribution, there might be some balancing act between protecting those who are like at high risk for death or bad outcome versus vaccinating those who are transmission vectors. You know, people who live in clustered environments or that sort of thing. Does your answer, does these recommendations, say from the National Academy, sort of balance that out, or are they optimizing according to one utility versus another?
That's a great question. I think the vaccine here, the vaccine is basically from being like protecting yourself, right? And so this should be considered different type of vaccine. So the other vaccine is how can one get the vaccine that preventing a case infecting the others. I don't think the current Pfizer vaccine and also Moderna vaccine is for that purpose. So the current Pfizer and the Moderna vaccine is more protecting yourself.
Hmm.
Yeah, and also if you know that if you see the data, and I don't have the slides here, the data are really impressive. And so, for example, like so I can I can read the data. So like for the Pfizer data, so totally there were about 44,000 people enrolled in the trial, and 170 people were infected. And then in the vaccine group, only 8 people were infected. In the placebo group, 162. And then, but only 1 person became severe in the vaccine group, and 9 people became severe in the placebo group. And in Moderna group, they were 30,000 people enrolled in the trial, and totally about 11 people were infected in the vaccine arm. 185 were infected in the placebo arm, and in the vaccine arm, nobody became severe. In the placebo arm, 30 became severe. So the data are really impressive.
Yeah, I think we're going to be seeing them coming out soon, even in peer review.
Mm-hmm. Yeah, I'm sure that will come out some in top journals, I would expect. Yeah.
We have a question from Donghui Yan. Donghui, are you there?
Oh, yeah. So I have a slightly technical question. So
Okay. Maybe just tell us where you're coming from too, please.
I'm from UMass Dartmouth. So the question is about this, the data used in calculating the reported cases, the ratio of reported cases out of all infected cases. So I'm just curious what data was used, or the data to which date, because I thought it's this ratio is might be decreasing. I thought.
So that you're talking about the ratio of reported cases to total cases.
Yeah. So basically, I'm referring to the tip of the iceberg figure.
So you mean this one, right?
Yeah.
So this red is the reported cases.
Okay, yeah.
And so like in US, and so we also analyze the US data. I just don't have time to present. And also we analyze Rhode Island data as well. We estimate the uncertainty rate in Rhode Island too. And so here, those reported data basically is the red part.
Oh,
and,
okay. So okay.
Okay, and so this yellow part, these are unascertained cases. Those were estimated from the partial differential equation Poisson model.
So what's your estimate about the US data? So I'm just curious.
The US data, I think there are a few things, and so the US data is different from Wuhan data. So if you look at the Wuhan data, you can see the data pretty clean, right? So not a lot of variabilities. But US data and the data are much more noisy. And so if maybe I can see, I have this, maybe use this. This is Rhode Island data. You can see the data are much more noisy and in Rhode Island compared to Wuhan data. So what we found is we cannot use the Poisson regression. And when we model the US data, we need to use over-dispersed Poisson. So we use the negative binomial distribution when we analyze the US data. And also the other thing we did was because in Wuhan data, the number of the test has not changed that much during that 2-month window, because it's very early on in February and March, there were not a lot of tests available. But in US data, the number of tests has gone up quite a bit in the last few months, right? And so when the number of tests going up, and then the uncertainty rate should go up too. And so we also model the uncertainty rate as a function of the number of tests and also positive test rate.
Yeah. So do you remember the ratio in US?
I don't remember.
Oh, okay.
Yeah, but we analyze the data for the whole US data, and also we also analyze by each state as well.
Oh, okay. So yeah. So I did some very rough analysis. So my estimate would be something like a 40% by end of April, and late August it's like a 20%. So something like that. So I'm just curious.
Oh yeah, I think we found that really vary by state, and because like for some state the number of tests has going up really well, and especially in Northeast, like in Massachusetts, for example, and I think for Rhode Island as well, the number of tests has gone up in the last few months. But in some state, for example, like in South Dakota, North Dakota, the number of tests has not gone up that much, and so therefore the ascertainment rate vary by state.
Oh yeah. Sure. Yeah.
Okay.
Thank you.
Thank you for that. Because I think the the lack of ascertainment, so the varying unascertained cases is just a huge driver of a lot of what we might be seeing.
Yeah.
Can I just also just a little bit of a sidebar before we get to the next question? In Rhode Island and in Indiana, there were statewide seroprevalence surveys. Do these kind of data feed into your model at all?
Yeah, that's a very good point. So what we did when we analyzed the US data was, because we need to have one point to calibrate the ascertainment rate, right? So in the Wuhan data, we use the flight data from Wuhan to Singapore to estimate the ascertainment rate in January. So as long as we have one point to calibrate ascertainment rate, then we can estimate ascertainment rate for the other time point. And so in the US data, we use the seroprevalence, the antibody rate, and use that point as to calibrate under the ascertainment rate. And then for example, like suppose like in Minnesota, for example, they did antibody studies, seroprevalence studies, and in April. And so we use we use that data and to calculate ascertainment rate in April. And so basically we have this kind of curve here. And so for example, think about this is April. And then we have this red part for Minnesota. And also we use the seroprevalence to calibrate what is the ascertainment rate in April. Then use that, we can estimate the whole curve.
I see. Okay.
Yeah. If for Rhode Island, so do you know how the sampling was done in Rhode Island for the antibody study?
Yeah, we did statewide random sample of households.
Oh, wonderful.
Yeah.
What is the prevalence did you estimate from that?
Well, we actually just turned in a revision of the paper. If you can believe, they're still asking for revisions. So they asked us to do an unweighted estimate was 2.2%, and then we did an age-weighted estimate, which turned out to be closer to 3%.
Oh, this is very similar to Wuhan. You can see that's very similar to Wuhan.
Yeah, that was the middle of May, actually.
Right, but here Wuhan, we did not have the seroprevalence data. This is fully from the PCR test data.
Right.
Yeah, from partial differential equation. So that is great to hear. Yeah.
Okay, so Ilana Green has a question. Ilana, are you there? And just a reminder to stay.
I am. Okay, so I'm an epidemiologist, and I'm actually at the Stat Center at Brown with Joe and everybody else. And my question is, the CDC has come out with new guidelines on the amount of time to quarantine after positive, and I was wondering if you have thoughts on that and whether or not your model can be used to address the effect of the changing guidelines.
Yeah, so that's a great question. And we don't have that data to evaluate that policy, because for that type of data, one needed to have individual level data. We don't have individual level data, so we only have the aggregated data. And so basically so this period here, the latent period. Here, this is about 3 days. And then from the pre-symptomatic onset to the symptom onset, this is about 2 days. And so in this period, so even though people, the case may not have symptom, but the case could still infect the others. And also the PCR test accuracy is not that great here. And the PCR test accuracy is great here. And so basically, if one calculates, you probably know the CT value. And so for the CT value, generally it is high here in the first week. So basically in the first week, it's most infectious. And so maybe I think the CDC may have individual level data, and then that makes them decide that probably after 10 days, and then the CT level is high enough. Generally, if the CT need to be less than 20, less than 30, and then that means a person is generally infectious. If the CT greater than 30, generally the person is not infectious. And so probably the CDC has a CT measures over time, and then they found out probably after 10 days, the CT value generally is greater than 30, and then they decided probably after 10 days, if somebody has a negative test, and then one doesn't need to be quarantined or isolated. So yeah, I don't know whether they have published any paper on reporting the data.
Yeah, I just saw the headline, so I'm going to look into it, but thank you.
Yeah.
Okay, so the floor is open for other questions. I think, Tom, you said you had a slightly more technical question.
Yeah, out of curiosity, how do you go about in your MCMC algorithm? How do you solve the differential equations, or how does the algorithm deal with the differential equations?
So we use the MCMC software. So there is a very nice software, and then one could use. So we did not write the code by ourselves.
Okay.
So basically, you just enter because it's MCMC, so that's why it makes things easier and to solve the differential equation while fitting the Poisson model.
Is
Okay,
this
so
software that we can all get our hands on, or is this like sitting somewhere at MIT or something like that?
So what software is this? Do you remember, or do you have a? Sometimes
what package we use?
Yeah.
I can send Joe the package we use.
Excellent.
And then Joe can forward to you.
That's what I was asking too. Yeah. Okay.
Yeah, because if we're talking about anything that doesn't have a bilinear system, then you can solve it explicitly. It's going to be a hypothesis of exponential equations, and you're home free with the MCMC. But here you have bilinear terms, and therefore you really have to do it numerically.
Yeah, and also we also have the code available in the Nature website, and so you could and also the data available there as well. So you could download the data and also run the code.
Now, in terms of the time series of data, is it only cases that you're using, or also hospitalizations and deaths?
So the data, as I mentioned, the data, these are the data. This is basically the reported cases.
Right.
The cases are the data. Those are the so in the Nature paper and also in JAMA paper, we only analyze the number of cases. But in JAMA paper, we also analyze the severity outcome as well, besides the number of cases.
Thank you.
All right, so we're coming to the top of the hour. I'll ask if there maybe is one more question out there for Xihong. It's really good so far.
I mean, since we have you here, and I don't know when we will have you again. And I throw
I'm
a last
happy
one.
to visit Brown again.
When Alison asked about whether you could use this model to explore different vaccination strategies, really, in order to explore different vaccination strategies, you would have to build in some memory into the model, in the sense of you want to know who got the first dose of the vaccine, who got the second dose of the vaccine, etc. So you cannot really use this model per se, right? You would have to build the memory in.
Yeah.
And the typical thing would be to expand the state space, put new compartments, etc. How would you do it? I mean,
so that is a good question. So you can see, like in this model, we did not put in any covariates, right? So we divided the data. So basically, we divide the data into 5 different periods, and then for each period we have the parameters, and then we fit all the data together. But we did not allow any covariates. We just allow the parameters are time varying for each period. But one could build in the regression model. So for example, here you can see we have no regression model. So one could build in the regression model to see how each of the mean depends on the covariates. And for example, if one has the individual level data, then depends on the covariates such as essential worker, healthcare worker, age, and so on. But we have not done that. Definitely, that will be a good topic.
thank you.
Yeah.
Okay.
Could I make an announcement, Joe?
Yes, please.
Okay.
Go ahead.
Okay. Okay. This is an announcement I'm going to make. So the COPS is called the Committee of Presidents and the Statistical Society. It has 5 charter members, like American Statistical Association, the East North American region, and the West North American region of International Biometric Society. And this is MS. This is the Institute of Mathematical Statistics. This is Society of Statistical Society of Canada. And also NIST is the National Institute of Statistics Science. And so the 7 organizations launched this new COVID data science webinar. And so the first webinar is tomorrow. And so that is at noon tomorrow. And so this webinar will be bi-weekly. It's all Zoom webinars. And so it's on Thursdays, every 2 weeks at noon. And so tomorrow will be on vaccine. And so we have 3 wonderful, wonderful speakers, and David Bankser, and from Emory, and also Beth Halloran, and from Hutch. And so they will talk about vaccine. And Natalie Dean from University of Florida, she will serve as a moderator. So this will be a great session. So if any of you are interested in vaccine and data, and so you are welcome to join the webinar tomorrow.
Thanks, Xihong. We can see if we can get this advertised on our DSI website for sure.
That would be great. I'll send you the link. It will be great if you can advertise it. Yeah.
Yes, I think we can do that.
Yeah.
We have a COVID data science page, such as it is. There's so much data science to keep track of with COVID that it's really hard to know what to feature. But this looks really good.
Yep.
So okay. So Xihong, again, I just want to say thanks so much. It's really been great to see you and hear about this really just remarkable trajectory of work and trajectory of analyses you've stitched together. And I think we're all really grateful for that. So I hope you're keeping well. I think a lot of us have to jump to other Zoom meetings, but if you don't mind staying on for 1 second, I'll just do some sort of last minute business to wrap up. But thank you everybody for attending, and enjoy the winter break, and we'll hopefully see you after that.
Thanks, everyone.
All right, so I'm in one of those, let's see, I think we have a couple of people who are still straggling here. Let me, okay. Hi again. That was great. Because I was thinking during this, like,